Hey everyone, so this is the lecture on chapter 23, the digestive system. I've cut down this PowerPoint, this lecture, in different parts because I know some of you said that my previous lectures were a bit long, so I've decided to cut this lecture up into different parts. So let's start with the first part of the digestive system, just some general characteristics of the digestive system. Just an overview of the digestive system. Organs are divided into two groups. So first you have organs of the alimentary canal. The organs of the alimentary canal include the mouth, the pharynx, and the esophagus. And then you have the lower part, which includes the stomach, the small intestines, and the large intestines. You then have accessory digestive organs. The accessory digestive organs include teeth and tongue, and then your different glands such as your gallbladder, salivary glands, liver, and pancreas. Now, the accessory organs are connected to the alimentary canal by ducts. Their secretions of these organs contribute to the breakdown of your food. So there are different ways of calling the digestive system. You can call it the gastrointestinal system. You can call it the alimentary system. Basically, the digestive system is just one long tube that goes from your mouth all the way down to your rectum and anus. And then you have the accessory organs or accessory digestive organs who have secretions or enzymes that help break down your food. This is a nice picture of the different structures that make up the digestive system. So you have the alimentary canal or the gastrointestinal system that goes from your mouth all the way down to the anus, just one long tube. And then you have the different organs that have secretions that secrete into the digestive system or the alimentary canal that, again, help break down food. So we know that our food is made up of three basic molecules. You have lipids, or fats, you have proteins, and then you have carbohydrates. So our body produces enzymes that break down these large molecules of food into monomers or smaller molecules of food. So for example, carbohydrates. Carbohydrates, another name for carbohydrates are polysaccharides. Polysaccharides, poly means many, saccharides, basically the structure of sugar. So carbohydrates are just complex sugars that we break down into smaller sugars or monosaccharides. And we have specific enzymes that break down these polysaccharides into monosaccharides. Proteins are proteins. We have enzymes that break down proteins in their simple components, which are amino acids. And these amino acids can be absorbed by the small intestine and by the first part of, specifically the first part of the small intestine, which is the duodenum. And then our fats, our lipids, they are broken down into triglycerides. So triglycerides, tri means three, so basically a triglyceride is a glycerol molecule and three fatty acids. So the idea of the digestive system is to chemically first break down our food using teeth, using enzymes, and then or mechanically, then chemically, mechanically using teeth, chemically using the enzymes that are secreted by our accessory organs so that they can be absorbed in the small intestine. We can divide the abdomen into regions. We have regions and we have quadrants. In order to divide the abdominal region, we have lines that can divide the abdominal into 9 regions. We had the midclavicular lines. We talked about midclavicular lines when we were locating the apex of the heart. So the midclavicular lines are the vertical lines of the grid. We have subcostal plane, the subcostal being that plane just underneath the costal margin. Costal means ribs. So the subcostal plane is the superior horizontal line connecting the inferior points of the costal margin. And then we have the transtubercular plane. This is the inferior horizontal line that connects the tubercles of the iliac crest. So the next picture shows you these different lines. So we have our mid-clavicular lines here, the vertical lines, and then we have the subcostal line right here, just below the costal margin, and then we have that intertubercular line here connecting the tubercles of the iliac crest. Okay, so subcostal plane, transtubercular plane, you can see here in this picture. Okay, so the superior one is subcostal plane and the inferior one is the transtubercular plane. And here you can see the 9 regions of the abdomen. We have different ways of dividing up the abdomen. We have our 4 quadrants, right upper, left upper quadrant, right lower, left lower quadrant. And then we have our regions. Surgically speaking, we use our regions if we want to identify a specific organ within that area, especially if we want to perform a surgery in that area, or if we're trying to clinically locate pain, abdominal pain. When a patient describes to us where the pain is, we can tell other physicians, oh, okay, this patient is experiencing pain in the right lower quadrant or the right iliac region. And usually we know that, the appendix is located in that area. So if you see this next picture actually shows you the different organs that are located in the different regions within the abdomen. So we can see, for example, in this upper region right here, which is called the right hypochondriac region. The right hypochondriac region contains the majority of the right lobe of the liver and part of the gallbladder. So the regions that you need to know include the right hypochondriac region. Here is the epigastric region, and here is the left hypochondriac region. And then in the middle we have the right lumbar region, because we know that the lumbar vertebrae are within that area. Then we have the umbilical region, where the umbilicus is located, or your belly button. And then you have the left lumbar region. And then the lowermost regions include the right iliac or inguinal region. We then have the hypogastric or pubic region and the left iliac or left inguinal region. Okay, those are the 9 regions that you need to know with regards to dividing the abdomen into the different regions. And of course, you need to know the lines separating the abdomen into those 9 regions. So again, regions are used to basically identify the organs within the abdomen so that you can localize any pain or localize a specific organ if you are performing surgery or any type of procedure. So dividing the abdomen into quadrants, we have 4 quadrants. Quad means 4. It's just a simpler method of sectioning the anterior abdominal wall. Again, we have our right upper quadrant, left upper quadrant, right lower quadrant, left lower quadrant. So these next slides kind of talk about embryology and how the abdomen develops. I'm not going to get too much into that. What I am going to talk about is how organs develop retroperitoneally, as well as I'll talk a little bit about mesenteries and how embryological structures become some of the more adult structures. So first I'll talk about the primitive gut. The primitive gut is developed through folding of the embryo during week 4 of development. So here is the primitive gut. Digestive systems or digestive system organs develop from the primitive gut. They are covered externally by visceral peritoneum. So again, think of peritoneum as that double-walled sac, just like the pleura of the lungs or the pericardium of the heart. So the peritoneum is the covering of the digestive system organs. So we have visceral peritoneum. Again, viscera meaning organs. So the visceral peritoneum is the part of the peritoneal lining that adheres to the actual organs. So our organs, digestive system organs in the peritoneal gut are covered externally by the visceral peritoneum. Then there's a peritoneal cavity that surrounds these organs, and then we have the outermost lining of the peritoneal cavity, which is the parietal peritoneum. So in this early embryo, all digestive organs are intraperitoneal, meaning they are surrounded by the peritoneal cavity. These organs are anchored to the dorsal and ventral wall by 2 layers of peritoneum fused to form a mesentery. So we have both parietal and visceral peritoneum fused together to kind of anchor these organs ventrally as well as dorsally. So we have a ventral mesentery here, anchoring the organs ventrally, and then a dorsal mesentery here, anchoring the organs dorsally. And these mesenteries, again, made up of the 2 layers of peritoneum that are fused. So both the visceral and parietal peritoneum are fused to form a mesentery. Okay? So mesenteries, these structures are passageways for blood vessels, lymphatic vessels, and nerves that supply the digestive system organs. Again, they anchor these organs within the peritoneal cavity. They also store fat, and they house the lymphoid tissues that will respond to any ingested pathogens. Now, as the digestive tract elongates and rotates, some organs will get pushed against the dorsal body wall. So the dorsal being the back, these organs will get pushed towards the back of the body wall. So the visceral peritoneum of these organs will then fuse with the parietal peritoneum along the dorsal body wall. So here you see this organ being pushed back towards the dorsal wall, and then the visceral peritoneum will then fuse with the parietal peritoneum along the dorsal body wall. At this point, the organs will lose that mesentery and then become located behind the peritoneal cavity. So here's the peritoneal cavity, and you see that this organ becomes what's called secondarily retroperitoneal in location. So again, these organs originally they were located in the peritoneal cavity, but then as with development of the fetus and development of this embryo, the organ gets pushed back and becomes secondarily retroperitoneal. There are organs that, such as the organs of the urinary system, that develop outside the peritoneal cavity from the very beginning. So, for example, organs such as the kidneys, we have the ureters as well as the adrenal glands or the suprarenal glands that sit on top of the kidneys. These organs were retroperitoneal from the very beginning. So this slide just shows you the remnants of the ventral and dorsal mesenteries and what they become in adult structures. So for example, the ventral mesentery will eventually form the falciform ligament of the liver, as well as the lesser omentum here that kind of has a connection with the liver and then part of the stomach. The dorsal mesentery, we can see becomes the transverse mesocolon, so this structure up here then becomes part of that greater omentum, which if you ever get to see the cadaver is that apron-like structure kind of anterior to the abdominal organs. Dorsal mesentery also becomes part of the sigmoid mesocolon, which you can't see in this sagittal section. Okay, so this is actually a really nice sagittal section of the contents of the abdomen as well as the different mesenteries. Below, you'll see the different transverse sections. So a higher transverse section at the vertebral level, T12. You can see, the liver right here. Here's the stomach. And then within the lesser omentum, which is that part of the mesentery off of the stomach, you'll see some vessels located within the lesser omentum. And then you'll see parts of the visceral peritoneum, which is the part of the peritoneum that surrounds the organs within the peritoneal cavity. And then the parietal peritoneum, of course, is the part of the peritoneum that lines the abdominal wall. Now, transverse section through the abdomen at vertebral level L2, you can actually see some of the retroperitoneal structures. So, for example, if we know that this is the parietal peritoneum and then going towards the dorsal wall, you can see part of the kidney. There's a section of the duodenum that's actually retroperitoneal. And then, of course, part of the pancreas is also retroperitoneal, meaning it's behind the peritoneal cavity. So the peritoneum, again, is a serous membrane, just like we have the pericardium as well in the heart and the pleura with the lungs. So we have the visceral peritoneum. This is the part of the peritoneum, or that layer that surrounds and adheres to the digestive system organs. And then we have the parietal peritoneum. This is the part of the peritoneum that will line the body wall. And then the cavity, the peritoneal cavity, is that slit-like potential space between the two layers. Mesentery is just a double layer of peritoneum made up of the visceral and parietal peritoneum. Mesentery helps hold and anchor the organs in place, also is a site for fat storage, and can provide a route for circulatory vessels or blood vessels, as well as nerves. Adult structures, so ventral mesenteries form the falciform ligament. This ligament it binds the anterior part of the liver to the anterior abdominal wall. Ventral mesenteries also become the lesser omentum. The lesser omentum attaches the liver to the lesser curvature of the stomach, and we'll describe the different curvatures of the stomach when we get to the digestive system organs. Then we have dorsal mesenteries. The dorsal mesenteries will become the greater omentum. This is that fatty apron that you see when you open up the abdomen of a cadaver. The greater omentum connects the greater curvature of the stomach to the posterior abdominal wall. The transverse mesocolon, this holds the transverse colon in place. And then we have the sigmoid mesocolon. This connects the sigmoid colon to the posterior pelvic wall. So again, just a common theme, these structures, these mesenteries basically anchor these organs within the abdominal cavity. So they're just not floating around in the abdomen. They're actually anchored by structures, remnants of the mesenteries. This picture shows the liver. You can see this ligament right here, the falciform ligament. And then you can see the ligamentum teres, which is, or also known as the round ligament coming off of the falciform ligament. And we'll talk about the embryological significance of the ligamentum teres in a bit. And then we see that fatty apron being a remnant of the dorsal mesentery. So this is the greater omentum. I believe a couple years ago they declared that they discovered a brand new organ and said, I think the greater omentum was this brand new organ. And technically speaking, the greater omentum has been around since humans have been alive. It's just they categorized this structure as a new organ versus just some, random structure. So it's not a brand new structure. We've always had one. It's not something that we never knew existed. It's always been there. It's just, it was categorized as a new organ. We can reflect back some of these organs to get to, for example, the lesser omentum, which is attached to the lesser curvature of the stomach. And then we have the greater omentum attached to the greater curvature of the stomach, which will hang down anteriorly to the intestines. So here is that greater omentum. And then we also have the transverse mesocolon, which attaches to the transverse colon of the large intestine. And then here we see the different mesenteries if we cut out the different parts of the small and large intestine. Again, these mesenteries anchoring the greater or the large and small intestine to the abdominal wall. So they're not just floating in space, they're actually fixed in a certain space. And we talked about the retroperitoneal organs. These are the organs that are located behind the peritoneum. There are structures that begin retroperitoneally. Again, these are the organs of the urinary system, the kidneys, the ureters, as well as the adrenal glands, also known as the suprarenal glands. These are the glands that just sit on top of the kidneys. And then peritoneal organs, these are the digestive organs that keep their mesentery and stay within the peritoneal cavity. So secondarily, retroperitoneal organs, again, initially they are formed within the peritoneum and then eventually become retroperitoneal. They fuse to the abdominal wall, their visceral peritoneum fusing with the parietal peritoneum and becoming fixed to the dorsal wall. And this is just a nice table that summarizes the intraperitoneal and secondarily retroperitoneal digestive organs in the abdomen and pelvis. Notice that the intraperitoneal organs include, so these are the organs that remain within the peritoneal cavity. We have the liver, the stomach, parts of the small intestine, the ileum, the jejunum, parts of the large intestine, so the transverse colon, as well as the sigmoid colon. And then we have our secondarily retroperitoneal organs that don't have mesenteries. This includes the duodenum, almost all of the duodenum, the ascending colon, the descending colon, rectum, and the pancreas. Now, there is a mnemonic that helps you remember the retroperitoneal organs. And you might see this again, possibly as a bonus question. So sad pucker. Sad pucker is a great mnemonic device that helps you remember the retroperitoneal structures, which include both primary and secondarily retroperitoneal GI structures. So sad pucker, S stands for suprarenal or adrenal glands. A stands for the aorta and the inferior vena cava. D stands for duodenum. The duodenum, the second through the fourth parts of the duodenum are considered retroperitoneal, whereas the first part, so there are 4 parts to the duodenum. The first part is still within the peritoneal cavity. The second through the fourth parts are considered retroperitoneal. The pancreas, all parts of the pancreas except for the tail, is retroperitoneal, both ureters, right and left. And then we have parts of the colon, so C stands for colon. We have the descending and ascending colon are retroperitoneal. K stands for kidneys, both kidneys, right and left. The esophagus, the lower 2/3 of the esophagus is retroperitoneal. And then finally R, the rectum, or a part of the rectum, is considered retroperitoneal. So you may or may not see this again if I ask you as a bonus question. So digestion includes important processes. So the first process we'll talk about is ingestion. Basically, you're putting stuff in your mouth, so ingestion occurs in the mouth. Propulsion is just the movement of food, and we have a specific term for the movement of food. It's called peristalsis. So peristalsis is the major means of food propulsion within the digestive system. Then we have mechanical digestion or mechanical breakdown. This prepares food for chemical digestion. It involves chewing, churning food in the stomach, as well as segmentation. Segmentation is just a rhythmic local constriction of the intestine to kind of move food around and allows for the increase of surface area for absorption. Next, we have chemical digestion. This is when we have complex molecules such as enzymes that break down food into its smaller components, and we can find enzymes being secreted in the mouth. We have salivary glands that secrete salivary enzymes. In the stomach, the acidic environment of the stomach helps with the breakdown of food. And then we have the small intestine, where we have secretion from the pancreas, from the hepatobiliary tree that secretes bile, also helps break down our food into smaller components, so that we have the next step of the digestive process, we have absorption. So once these molecules are broken down both mechanically and chemically into their smaller components, they then can be absorbed. The majority of absorption of our food occurs within the small intestine. So absorption is just transport of digested nutrients into our system, so that we can use these molecules to produce ATP, to provide energy for our tissues. And then the last digestive process is defecation, pooping. So elimination of indigestible substances as feces. Example I like to give is corn. So corn, we all love corn, especially during the summer. I'm from Jersey. We have the sweetest corn, in my opinion. Corn is a complex carbohydrate. We don't produce the enzyme to break down corn. That's why we see pieces of corn in our poop, in our feces. So this is considered indigestible because we do not have the enzyme to break down this complex carbohydrate. The next slide just shows the activities of the gastrointestinal tract. So we see the different processes that occur within the digestive system. We have ingestion of food into our mouth. We then have the different breakdown of our food. We have mechanical as well as chemical breakdown. So we have chewing that occurs in the mouth using our teeth. Churning within the stomach also helps break down food mechanically, and then segmentation within the small intestine also helps break down food. Other ways that we break down food is through specific chemicals that are secreted into the GI, such as our enzymes, as well as the acidic environment within the stomach, breaking down our food into smaller molecules. Propulsion, this is just movement of the food within the digestive system. So we have swallowing occurring in the oropharynx, that second part of the pharynx where food is then moved into the esophagus. We then have peristalsis, just the fancy word we use describing the movement of food within our GI. So peristalsis, of course, occurring in the esophagus, the stomach, small intestine, large intestine. So we have that layer of smooth muscle lining these structures that we saw in our histo slides earlier this semester. So that smooth muscle helping with peristalsis, or the peristaltic movements of our food throughout our GI. Once we are able to break down our food into smaller molecules, we then have absorption into blood vessels so that these smaller molecules can travel to our tissues, allowing for metabolic processes to create ATP, to build up tissue. So we absorb our food so that our body can use it. And then finally, our last process in our GI system, we have defecation. So we remove any indigestible food that just we can't use. So peristalsis is a major means of propulsion. Adjacent segments of the alimentary canal will relax and contract to help move food throughout this tube. So adjacent segments of the alimentary tract organs will alternatively contract and relax, moving food along the tract distally. And of course, we have that layer of smooth muscle within our gut to aid in this movement. So segmentation is just a rhythmic local contraction of the intestine, also mixes food with digestive juices, helping with further digestion of food. So with segmentation, non-adjacent segments of alimentary tract organs will alternatively contract and relax, moving the food forward, then backward. Food is mixed and slowly propelled. And just quickly talking about the histology of the alimentary canal wall. Same 4 layers are seen from the esophagus to the anus. So we have the innermost layer known as the mucosa, consisting of epithelium, lamina propria, as well as muscularis mucosa. We have then the submucosa, which is outside or external to the mucosa, that contains blood and lymphatic vessels as well as nerve fibers. The next layer is the muscularis externa. This is outside or external to the submucosa, made up of 2 layers. So we have an inner circular layer, also known as the circular muscularis. This is that inner layer. And then we have an outer longitudinal layer, so the outer longitudinal muscularis layer. These layers are layers of smooth muscle, again, that help move food within the digestive system. And then we have the outermost layer. This is the serosa. This is that visceral peritoneum that adheres to the digestive system organs. So here we can see the different layers of the alimentary canal. We have the innermost layer, the serosa, made up of epithelium as well as connective tissue. And then we have the subsequent outer layers. Important to note are the different muscular layers. So we have that inner circular layer made up of smooth muscle, and then an outer longitudinal layer. So basically, the smooth muscle is oriented, the inner layer being a circular layer, and then the outer layer being a longitudinal orientated layer of smooth muscle. And just a histological slide of the alimentary canal, so we can see the innermost mucosa. Then we have the submucosa, which contains, lymphatic vessels, blood vessels, nerves. And then we have the muscularis externa with that inner circular layer and then outer longitudinal layer. And then we have the outermost serosa, which is the visceral peritoneum. So again, smooth muscles primarily found in the walls of viscera. Fibers are elongated, have one centrally located nucleus. And smooth muscle, we know, differs from cardiac and skeletal muscle in the fact that they're not striated. Smooth muscle in the digestive system, again, in the muscularis layer, are grouped into sheets. So you have a longitudinal layer. This is the outermost layer, and it's parallel to the long axis of the organ. And then we have that innermost circular layer of smooth muscle. This is the deeper layer where the fibers will run around the circumference of the organ, forming a circle. So here you just see a cross-section of that muscular layer. So we have that inner circular layer of smooth muscle, and then an outer longitudinal layer. We talked about contraction of muscle earlier in the semester. So we know that myofilaments operate by interaction with the cytoskeleton. We have dense bodies that correspond to Z discs of skeletal muscle. And then we have sliding myofilaments that shorten the muscle cell by pulling on the cytoskeleton, and we have that important ion, calcium. Entry of calcium into the sarcoplasm will stimulate contraction, as with skeletal and cardiac muscle. So calcium, again, being that very important ion that helps stimulate contraction of muscle. So with smooth muscle, contraction is slow and sustained. It takes about 30 times longer to contract and relax. However, it is resistant to fatigue. Smooth muscle of arteries and visceral organs must sustain contraction over long periods. Energy requirements are low, so with smooth muscle, mitochondria are not abundant, not as abundant as, say, skeletal muscle or cardiac muscle that need more energy to perform their actions. And here we see a smooth muscle fiber that's both relaxed and contracted. Up here we'll see a relaxed smooth muscle fiber. There are gap junctions that connect adjacent fibers. So here are those gap junctions. And then here we see a contracted smooth muscle fiber. Smooth muscle is innervated by the autonomic nervous system. Few fibers per sheet are innervated. A sheet of smooth muscle contracts as a unit called a single unit innervation. With multi-unit innervation, each smooth muscle cell is innervated. Examples of a multi-unit innervation within smooth muscle includes the iris of the eye, as well as the erector pili muscles, those muscles that we saw in the skin that help the hairs in your skin kind of stand up on end. For example, if we're feeling cold or if we're feeling scared, like if there's a ghost in the room, or if you forget that you have an exam and all of a sudden the exam's that day. So here we just see innervation of smooth muscle. We have our autonomic nerve fibers that innervate most smooth muscle fibers. And then we can see the different varicosities that will release neurotransmitters into a very wide synaptic cleft. And within these varicosities, of course, are mitochondria that help produce energy. We have different nerve plexuses that will innervate our smooth muscle. We have the myenteric nerve plexus that lies between the circular and longitudinal muscularis layer. This helps control peristalsis as well as segmentation within the GI. And then we have our submucosal nerve plexus that will lie in the submucosa, helps to signal glands to secrete. Innervation of the digestive system, we have sympathetic and parasympathetic motor fibers, as well as visceral sensory fibers. So remember, sympathetic nervous system being responsible for our fight or flight responses, usually has an inhibitory effect on our digestive system, whereas our parasympathetic fibers, they are responsible for our rest and digest. So parasympathetic motor fibers actually stimulate the digestive system to help stimulate peristalsis as well as digestion, and have opposite effects compared to the sympathetic nervous system, because it makes sense. If we want to run away from a dinosaur, we don't want our digestive systems working. We want, other things working. And with our parasympathetic nervous system, this is the time where we can digest food, where we can, helps us with resting our bodies. So it makes sense that, sympathetic will have a negative effect on the GI system. And that's it for part 1 of the digestive system. Part 2, we'll start going into the different organs.